Looks perfect.
Okay. All right, shall I get started?
Yeah, go ahead. Thanks a
All
lot.
right, thank you. All right, well, it's, again, a pleasure to be here and to share our work on molecular height measurements by cell surface optical profilometry. So I'm going to give just a short introduction and motivation for the work, and then I'm going to turn it over to Dr. Sungmin Son, who was the first author on the paper and developed the technique. So by way of background, I want to point out that the popular view of plasma membranes is lacking in many respects. While it's true that it's a lipid bilayer, this popular image from a Google image search suggests that the features of membranes that we want to focus on today aren't necessarily part of that popular view. And in particular, we want to talk about the diversity of sizes of ectodomains of cell surface proteins. And this picture is missing them, but they can range from a nanometer to hundreds of nanometers. And they involve a diverse number of distinct species as well. About 3,000 different proteins in the human proteome can be found on cell surfaces. Now, we know from structural studies that the cell surface proteins can have a variety of sizes. If you simply look at either solved structures or if you add together the domains that make up an extracellular domain, it's clear that they must exist over a range of different heights. We also know that for many years from electron microscopy, which has captured the thick glycocalyx that can be found on some cells. So it's not just the proteins, but it's the glycans on those proteins and that create the size and create a very complex interface that other cells must interact with. We've recently been exploring that diversity by looking across the human proteome and estimating, based on homology modeling, what the sizes of cell surface proteins are. And so this is work by a postdoc, Caden Southard, that shows quite a range of different protein sizes, predicted protein sizes, as well as some interesting peaks in the distribution that appear to correspond with multiple domains. But understanding exactly the role for the different heights is something that prompted this work. As is clear from thinking about any cell-cell interface, the ability to engage one cell with another, to transfer information, to mechanically couple two cells, requires adhesions and requires molecular interactions that must take into account the diversity of sizes of those molecules, both the molecules involved in the binding as well as the other molecules that are present on the cell surface. And so that density and that diversity raises the question of what happens when these two diverse surfaces come into contact. This has been a question of interest for many years, going all the way back to the original Bell model for thinking about how a bond under force is going to have a reduced lifetime. And that has been extended to thinking about if the resting separation between two things that can bind is greater than their natural length, how is that going to affect their on rate? So the ability to engage is also going to be influenced by the gap created between receptor and a ligand. And so there's a great deal of theoretical understanding about what influence molecular size can have on receptor binding. 30 years ago now, Tim Springer, in a wonderful review article that mapped out the different sizes of immune receptors and adhesion molecules, really, I think, brought forward the idea that the different sizes are probably going to need different gaps between membranes to exist, and that because you have small and you have large, that they are likely to be spatially segregated. And in the context of T cells, the kinetic segregation model was proposed in 1996 by Anton van der Meer and Simon Davis, which really focused on how potentially the size-based segregation of a tall phosphatase could be responsible for or contribute to activation of T cell receptors. And so this is a really interesting hypothesis, mechanistic idea that has borne out over the last couple decades. We initially got interested in this question from a very in vitro perspective. And the question was, how big do you need to be influenced by the gap created between two membranes? How tall must a protein be to be segregated? And we explored that problem in the context of giant unilamellar vesicles. So this is work from Eva Schmitz and Matt Bacalar. And what they found is that you only need to be about 5 nanometers taller than the gap in order for the gap to have a marked influence on where you are localized. So it doesn't, the membrane is not able to simply accommodate different sizes. The membrane at an interface will drive segregation of things that are too large. And only 5 nanometers, just the size of an IG domain, is sufficient for that exclusion. We then became interested in whether that could have a role in the context of macrophages. As a macrophage, as an immune cell comes into contact with a target, how does the complexity of the cell surface influence the engagement of the key receptors needed to activate macrophage activity? And the activity of most interest was phagocytosis. So for a foreign object or an object that is labeled by an antibody, how does the macrophage know that this is the right object to phagocytose beyond simply the handle of having the antibody on its surface? And so what Matt Bacalar and several students from lab were able to figure out was using a semi-synthetic system, a glass bead coated with a lipid bilayer, and by attaching multi-domain proteins, synthetic multi-domain proteins, that had an epitope for an antibody at the very end, they were able to show that even if you keep the density of antibodies the same on a particle, if you simply extend the height at which that antibody is presented, so essentially the antigen height, you can dramatically reduce and even shut off phagocytosis. So long molecules, if you have an antibody at the tip, are very poorly phagocytosed. And this has everything to do with the kinetic segregation model that I described earlier, in that the size of an inhibitory phosphatase is important for this process, that it needs to be excluded in order for the FC receptor to signal. But when we were making these multi-domain proteins, these fibronectin domain repeats, the question was, well, we added a domain, but how much actual height had we added to this protein? And that's when Sungmin and Matt devised a very clever method for figuring out how tall is this protein by attaching a fluorophore on one end and on the other end, and then localizing both fluorophores. And that was really the motivation for the CSOP technique, the ability to measure directly the sizes of cell surface proteins is something that we couldn't do with the nanometer scale precision, but wanted to do. And we wanted to do it for that macrophage experiment, but also in general. So here's where I will turn it over to Sungmin to describe how he took this challenge of making a nanometer scale measurement and turned it into a new technique.
Thanks, Dan. Sungmin, could you also introduce yourself with a few sentences before you start?
Okay, so a bit of about myself. My background is precision measurement and instrumentation in PhD. And in Dan's lab, I've been working on two mainly different areas. So one is about virus entry. More specifically, I'm studying how those dense proteins on the surface of virus and the cell will present the physical barrier of the virus membrane fusion. And that was the pre-COVID times. But during the COVID times, I'm working on making a rapid COVID detector. Okay, so share my screen and go from there. Okay, so like Dan just introduced, proteins on the surface of cells come in very different sizes, but functionally, even a few nanometer change of those sizes can change the signaling and the way that interfaces are formed. So I took this measurement challenge of how to precisely measure these targets that are vastly different in size. So before going further, let's start by reviewing some of the imaging techniques that's available for now. Usually for the live cell, confocal microscopy is giving very clean, very nice images. But systems like this is diffraction limited, so you can't resolve the structures as finer than a few hundred nanometers because of this physical diffraction limit. And there's very clever technique called super resolution microscopy, which statistically overcomes this diffraction limit, and you can achieve as good as tens of nanometer resolution. And also there is a very specialized super res imaging where it allows the resolution of even 1 nanometer. But most common super res technique, their resolution is still limited by larger than 10 nanometer laterally, and vertically the resolution is much worse, like 50 nanometers. Okay, and most of the times these imaging techniques needs the cells to be fixated, and the imaging time is very long. For the resolution that is necessary for studying cell surface proteins, EM, electron microscopy, is a very good technique, although it's lacking molecular specificity. So while we can resolve these nanometer structures on the surface of cells, we might not know what the nanometer structure is actually from. And again, the preparation of the EM microscopy is very lengthy, and it's sample specific. Okay, so I saw some needs for a new tool to study this, and basically the idea of cell surface optical profilometry is a 1D localization, right? So let's imagine there is these 2 lines of different colors. So say membrane with green and the tip of the protein is labeled with red. If you use confocal microscopy to localize these planes, then what you see in the image will be a very thick line that is 200 to 300 nanometer thick and mostly overlapping to each other. But just like a conventional super res technique, you can try statistically localize the centroid of each lines, and precision of doing so will be much better than those diffraction limits. And practically, to localize those lines, instead of comparing these 2 flat lines, I found that arranging those fluorophore in a circle and basically estimating the radius and compare those radius to ask what is the offset between those radius is a much efficient way of comparing those offsets. Okay, so you can appreciate these by comparing these plots in the top and the bottom panel. So basically, just simple line skin will give you this Gaussian looking signal distribution, and you usually try to localize what's the location of this. But then if you're radially averaging those signals, your background noise reduces significantly while your signal remains the same. Right, so I used the expanding circle to ask how the fluorescent signal is changing over this radius. So basically, this signal to noise boost from the top to the bottom panel is what is giving the precise resolution of this technique. And just like other super res technique, this resolution is limited by how many photons we can collect. And so as we collect more and more photons, it looks like the resolution of these techniques can approach even below nanometers. So again, the difference is that the number of photons we can collect is not only limited by this 1-line scan, but how many fluorophores reporting the same locations, which is just simple dimension of R. So to make this comparison a little bit more intuitive, let's think about 2D localization techniques. So you can think measuring the distance between 2 different color labels in 2D. And again, you would localize this location of red and location of blue, but this localization accuracy is strictly limited by the photon budget of each fluorophores, and it's something that is hard to improve. So your distance measurement is limited. And in this particular study, they do some additional aberration to overcome these limitations. Okay, so with this principle in our hands, we wanted to validate this technique by using a molecular standards. And the easiest size standard is a double helix DNA, right? So we have this series of double helix DNA that one end is labeled with the red fluorophore and the other is tagged with the cholesterol. So I would create this lipid-coated piece where its membrane is doped with the green fluorophore. So it's marking the location of the membrane. And then I would add very small amount of each DNA into a well so this DNA can spontaneously incorporate to the membrane. So now I have, several beads that's mobilized on the surface that has red fluorophore and green fluorophore, which I can characterize a radius to measure the offset, which is my size measurement here. So this is one of my first data set, and I was very excited to see that, increasing this base pairs of DNA actually increases the height signal. But then upon closer examination, the number seems a little bit different than what I anticipated. Because DNA, it's very well known that the length of DNA is about 0.36 nanometer per base. And my measurement was actually exactly half of each one of those, right? So then now I started thinking about the actual situation of these fluorophores, and it must be wildly fluctuating while the DNA molecule is bending, rotating, and swinging on the surface of this membrane. And as I simulated the tip locations as it does that motions, the center of mass of this tip location distribution was actually half of its maximal vertical height, suggesting that we are measuring not the fully extended end-to-end distance of DNA, but its average tip locations as it's exploring the whole different configurations. Okay, so it's pretty good. The principle still holds, but to actually make this accurate measurement, there was one very important calibration step. This is calibration for chromatic aberrations. So in the microscope, different light of different wavelength is bending differently as it transmits through a lens. So that's chromatic aberration. And in our case, it shows as different size of circles when you image the red and green circles, for example. And the size of this is actually different both in the lateral plane and also in the vertical plane. So to first predict the actual radius of two different red and green circles, you have to measure the midplane in different locations in z directions, right? So instead of taking just one snapshot in a kind of midsection of the beads, I'm taking a stack of images and then try estimating what's the midplane for both green and red color circles. And usually, the most intuitive way is to look at the distance between this red at the tip and green on the membrane. But because, again, this chromatic aberration in the lateral plane will give you some offset, so the size of these circles will look different. So that's where this calibration step is coming in. You need a target that the same plane is labeled with both red and green. So without chromatic aberration, the offset between those two radius should be 0. But because of that aberrations, I see some offset, which has to be simply subtracted from your actual protein height measurement. Okay. All right. So now we have the principle, we took care of the measurement method with calibration. So now we wanted to push the limit of our measurement. So in this case, we try to resolve the thickness of two different lipid bilayers composed of different acyl chain lengths. You know, measurements of lipid bilayers should be doable because lipid bilayer thickness is about 3 to 4 nanometers. But resolving the leaflet sizes composed of two different acyl chain lipids must be beyond the limit of this detection methods. But we thought maybe we can repeat the same measurement from the same sample over and over, then at least we can predict what's the average estimation of those thickness, and this might put us beyond the limit of resolution. So here's how we did the measurements. First, we need the red fluorophore that's always marking the midplane of this lipid bilayer. And to measure the thickness of one leaflet, we basically used cholesterol labeled with a green fluorophore. And I either incorporate those cholesterol in both leaflets, or I first made this supported lipid bilayer only with the red fluorophore, and then added through solution this cholesterol fluorophore that will decorate the outer leaflet of these lipids. Okay, so this is usually the distribution of our baseline after correcting for the chromatic aberrations. And you can see, comparing to this baseline, the leaflet marking green fluorophore's distribution is offset, positively offset to report the thickness of one leaflet. And quite satisfactorily, we are seeing the difference, some difference between two membranes composed of different acyl chain lengths. And this difference, although quantitatively might not be the most accurate, it is in the right trend, and the difference is in a similar order of magnitude with the previous measurement that's measured with the EM techniques. So there is a recent, very clever EM techniques who I believe gave a journal club 4 in this meeting as well. And then it predicts that, I mean, it really visualized that the thickness of these membranes are different, and the numbers are consistent with my measurement. Okay, so let's focus on the proteins actually. So we wanted to take the synthetic proteins that we used for the macrophage phagocytosis study and measure its size actually. And when we did that, there is an apparent deviation of this size from this 1/2 line, which would be the case for the perfectly stiff rods, right? So it's pretty different from the DNA, and the larger proteins are rolling off its average size from the stiff cases. So it was interesting, and Sho Takatori decided to model this using a MD simulation. So in this simulation, he basically modeled each protein domain as a hard sphere and introduced a rotational stiffness between each of those two domains. And then by tuning that rotational stiffness, he can recapitulate the motion of semi-flexible polymers. So the semi-flexible polymers, you can describe the entire motion by this one parameter called persistence length. And truly, when we actually change the persistence length, he predicted that the tip distribution will change drastically. And that comes with the change in the average height as well. So in our case, when the persistent length was about 13 nanometer, it's matched with the data most accurately. So it basically predicts that 13 nanometer persistent length is about 3 domains, and the implication of this measurement is that the orientation information of multi-domain chain proteins will be maintained up to 3 domains, but after that, they don't know what's the orientation of the first domains, right? Okay. So far, I've been measuring the proteins in a sparsely kind of spaced conditions. But on the real cell membranes, like Dan mentioned, the proteins are packed. They're presented at the 10 to 20,000 proteins per micron square. And at this packing, the protein-to-protein distance can be as close as within 10 nanometer, and tall proteins might fill each other. And it will create some crowding pressures. So we wanted to see if these crowding pressures will have an effect on the size of the total protein height. So that's why what I try to recapitulate it here. Increasing the binding concentration of those proteins on the surface, and I start to see that the average protein height I measure is actually start to increase. But there was a limit to how much I can add these proteins on the surface. So using an MD simulation again, we could see that this concentration range where it's more relevant to the physiological concentration of cell surface proteins, there is very significant change that the protein height has to go through. So it's basically suggesting this protein height is not a fixed parameter, but depending on the microenvironment and the crowding states, this height can adapt to different locations. And it's not only the lateral surface crowding, but also the solution crowding that can affect this protein average height, right? So in the blood, there is a high concentration of blood serum proteins that can work as an osmolyte that have an effect on the surface, and polymer brushes are well known to change its conformation because of this osmotic pressure. So in our case, we wanted to test this solution osmotic effect by adding a osmolyte. In this case, it's a PEC. And as we are increasing this PEC concentration, and most importantly, to the level that is resembling the proteins levels in blood serum, the height of proteins starts to decrease significantly. So it has an interesting implications. It's not only the glycophilics or the polymer brushes that really falls because of this high osmolarity, but even this tens of nanometer high proteins can change its height because of these solution conditions. Okay, so far I've been telling you about the data of synthetic protein measurement. So, here is the real proteins. I modified the tip of these proteins the same way though to label its protein tip, and the measurement looks qualitatively pretty similar. So the trend is that these 1 to 3 domain proteins looks pretty stiff, and if the protein length becomes more than 4, 5, 7 domains, then the average height seems to be lower than a stiff rod. And that's consistent with what I saw with the synthetic proteins. Right. But having to label each protein with the specific labeling method is pretty limiting the application of this tool. So we decided to explore if we can just use a labeled antibody, but tagging it to the unmodified proteins to report the height of these epitopes that antibody is tagging to. Right, so to test this idea, first I used a synthetic protein again. So now this protein at the tip is modified with the biotin. And I used labeled biotin antibody to report the location of these biotin epitopes. So in this case, this antibody is labeled in its exposed lysine locations, and because it's labeled randomly, I would anticipate that the location of the average height of this antibody will be its center of mass. So now I've already measured the tip average height of these synthetic proteins. Now I compare the antibody location bound to its tip with its measured tip height. And I could see that there is a very predictable offset, positive offset between the tip to the antibody. And using MD simulation, we confirmed that the only parameters that predicts this offset is the location of antibody height, right? Antibody can be bound in the middle of these tall proteins, but still the offset between the antibody location to its epitope will be just a function of antibody height. Okay, so with this kind of calibration curve, now we can use anybody's height measurement to back out what's the epitope locations. All right, so we have all these tools ready. So let's try measuring some interesting proteins. So now I am focusing back to what Dan has introduced, which is the tyrosine phosphatase, CD45, CD43, which is very important in signal activation of immune cells such as T cells as well as macrophages. The reason why the size of these proteins are very interesting is because they are segregated from the activating immune interfaces. And it's thought to do that surely because of this large ectodomain size. But ironically, it's been very challenging to actually measure the size of proteins because most of its ectodomains or half of its domains are unstructured and heavily glycosylated. And while the structure of folate domains are easily solvable and its size can be predicted by these crystal structures, these unstructured domains is very hard to predict. Okay, so I decided to use many commercially available antibodies against the CD45 proteins and try to measure the average location of these antibody epitopes. So this antibody has a isoform preference, or isoform specificity. So I have rough idea of where these antibodies are binding to, but I don't have a knowledge of exactly where on the length of protein is binding. So I have 4 different antibodies, and I have 2 pan antibodies that's supposed to bind to the folded domains of CD45. I also have 2 different versions of antibodies that will bind to the mucin-like domains that is unstructured on top of these folded domains. So what's convenient for us is CD45 has different isoforms, and different isoforms actually have different mucin-like domains length. And the longest of which is isoform called RABC. And it's expressed in the B cells, and this B220 antibody is named because it's targeting the 220 kilodalton version of the B cell CD45. So apparently, this has to be targeting the tallest location of the CD45, which is what we are seeing. And the other antibody is targeting the B isoform of the CD45 proteins is measured a little bit lower, while the other 2 pan antibodies are located very low. And because of the calibration curve, we can predict what would be the epitope height, which is marked in this red circle. And the trends make sense, and we could predict what might be the exact location of these binding epitopes. And we can do the same thing for the glycan measurement as well, because there are different lectins targeting different glycan with specific affinity. These CD45 proteins in the mucin-like regions, it's known that there's heavily it's heavily sialylated. So we anticipated that the lectins such as SNA that is targeting this mucin-like domains should come out as higher average height. And that is comparing to other lectins targeting galactose or galnet of the same proteins. Okay, and it was the same for CD43. So Sam, during her rotation in our lab, decided to take this measurement approach and measure the height of the tip of CD43. And interestingly, the tip of CD43 height was similar to the CD45's height measured by the B220 antibodies. And they're both about the average height of 20 nanometer. And again, if we look at this curve of increasing protein size, the full end-to-end distance of these heights can be as 40 nanometers if these proteins are perfectly stiff. And it can be even longer than 40 nanometers if these proteins are rather flexible. And it's kind of remarkable that this literature from 20 years ago predicted those height of these active domains quite accurately. Okay, but what we're measuring here is, again, its average height while these proteins are dynamically moving. And that might be a more meaningful parameters when we are thinking about the physical segregation models. But again, these parameters has to be considered in the context of other proteins. So we decided to characterize even more general protein heights in the surface of cells. So this technique, CSOP, required that the protein should be arranged circularly. And to measure the general protein height on the surface of a native membrane, we needed the membrane to look circular. And there are special techniques to achieve that. And Erding is actually an expert of one of those techniques. So here you can use this technique to make what's called giant plasma membrane vesicle. You can chemically treat the cell and cell becomes unhappy and it starts to develop a large patch of very circular membrane of its own. Another way, sometimes cells are not so amenable for this GPMB treatment, or GPMB might not drag all the proteins into its content. So in another realization of this, we can simply get rid of the actin cytoskeleton by drug treatment, and then hypoosmotically bloat the cells to a circular shape. Okay, so we first validated this height measurement to these different platforms. To do this, we used that CEACAM5 proteins and a 7-domain Ig-like domain protein. And we found that both the height measured in the GPMB or a swollen cell showed very similar numbers as the height measured in the beads. And that seems to be the same for other proteins as well. So I made GPMBs and measured 4 different kinds of my actual proteins. And the height was comparable between the glass beads and GPMB case, except for the case of SERB alpha. So we thought, oh, it was intriguing, and maybe, for example, CEACAM is a GPI anchored protein naturally. So it can freely swing back and forth while it's sitting on the actual plasma membrane. But SERB alpha has a transmembrane linker, and we thought that the membrane linking mechanism might make the protein stand more vertically. So we modified the proteins to the same way as CEACAM. So basically introduce GPI in place of the transmembrane domain and expressed in the surface plasma membrane, and then measure this height. Although we are seeing that the height is actually decreasing comparing to the wild type case, suggesting that the linking mechanism of to the membrane actually affects the average height of proteins. There seems to exist other factors that's really affecting some of the protein's average height in the local microenvironment. It can be glycosylation, it can be other surrounding proteins identity. Anyway, so moving on, we are interested in how do we interpret this tall CD45 protein's height in the context of other general proteins. So here, instead of focusing on one specific proteins, we decided to characterize the general protein height by promiscuously labeling its N-terminus with biotin. And I added the biotin to measure those general N-terminal height of proteins. And I could also do the same for 2 different kinds of glycan targeting lectins, right? So now we clearly see that the CD45 is significantly taller than either the average proteins or the distribution of glycans on those proteins. On the other hand, in this macrophage progenitor cells, HOXB8, the height of Fc receptor was significantly smaller than the average protein heights. So now it starts to kind of give the picture of how this membrane surface is looking like. There can be a specific toll protein such as CD45 that can be physically segregated, while this Fc receptor is trying hard to overcome the average protein height and establish an interface with the shorter gap. Right, and these macrophage is sometimes making a interface with the cancer cell. So I thought we'd test the cancer cell surface profile as well. So in this case, we measured the HER2 positive breast cancer cells called SKBR3. And again, the average protein height turned out very similar as the macrophage's average protein height, while the sialic acid distribution is a bit taller. And most interestingly here, the famous cancer antigen HER2 seems to be buried in this general protein height, because the trastuzumab, which is a therapeutic antibody that is targeting the membrane proximal domain of HER2, its average location is much significantly lower than the average protein height. So this pretty short, 10 to 20 nanometer of average proteins distribution in the cell surface is kind of picturing a little bit different things, kind of idea than the thick cancer glycocalyx. But it is consistent with the bioinformatic estimation that Caden was able to make. So if you filter the distribution of proteins expressed in KBR cells, then the N-terminus height is in average about 12.6 nanometer. So it's basically saying that although we know that in the actual breast cancer cells, there is this very thick, hundreds of nanometer thick glycocalyx that's presenting some physical barriers. Still, there is very thick, dense population of proteins in the 10, 20 nanometer distant from the cell surface that's presenting another physical barriers. And those has to be overcome by cells like macrophage to target this therapeutic antibodies binding deep into the surface of cancer cells. Okay, so this is what the demonstration of CSOP technique could do. And with this, I'd like to end by thanking the lab. A lot of members participated in the aspect of this study. And throughout the postdoc, Life Science Research Foundation funded my postdoc, and these are other funding sources. Thank you, and I'll take any questions.
Thank you very much, Sungmin. This is brilliant. Thanks very much, Dan. So I will start with my own questions, just because I can. So the first question, then, I want to ask you. So you previously showed that now, if from your in vitro measurements, if the protein height is 5 nanometer larger than the contact size, it's enough to exclude the protein. So from now, what we learned from the new technology, the distribution of the cell, of the proteins, the height distribution, what would you say about A, the size of the synapse? Should we revisit the size of the synapse? Second, this 5 nanometer larger than the contact, can we revisit this size? Is it now different size?
Yeah, great questions. I think it's probably worth reevaluating both. So I think on the in vitro measurements, I think it's important to keep in mind that that's in the absence of any other organizing principle. at an interface, I think there's going to be multiple influences. So it's not simply going to be size of proteins, but order in the membrane, connections to the cytoskeleton, actively driven locations of proteins. Many other influences can be responsible for the observed spatial organization. So I think what we had hoped to show in that in vitro set of studies on the 5 nanometer being enough to exclude was that in the absence of other organizing forces, that 5 nanometers is enough. And so I think it'll be interesting to know in the context of a real cell-cell interface, how big are the ones that are actually excluded? And that would then address the other influences, membrane organization, cytoskeletal connections that are at work as well. I think it's also interesting to think about the gap size. What is it really? How uniform is it? What are its locations? So potentially an extension of this technique that Sungmin described to measure interface gaps would be useful as a next step.
No, totally. I totally agree. So related to this real cell measurements, there's a question by Christopher Kelly. And he is asking, is it correct that this technique measures the average protein height while averaging over membrane undulations and lateral heterogeneity? Although you measure it in vitro or in GPMVs, how would this be applicable in the heterogeneous and moving dynamic cell membrane?
Maybe I'll turn this over to Sungmin to describe some of the measurements, some of the controls that he did, looking at both lipid bilayers on glass beads in comparison to lipid bilayers on freely fluctuating membranes, which might be the extreme of, or at least a passive example of a membrane.
Right. So basically, the question is whether these techniques will measure the precise protein height when the membrane underlying is fluctuating. So I compared the GUV and the lipid-coated beads and bound the same proteins on the height. And I could back out the same average height measurement regardless of which membrane it was binding to. And that's because while the membrane is undulating and it might bias the kind of rotation of the proteins, I guess it didn't actually bias the height distribution of the tip kind of motions by membrane undulation itself. And those undulations will be averaged out because I am averaging those radius from many, many photons of membranes anyway.
So in the systems that you measure, when you get the radial averaging, as far as I understood, you're basically increasing the radius of the sphere that you're measuring, and then you get the averaging. So if the object that you are looking at is not perfectly spherical, you still have a way to correct for this.
Right, so it depends. So say your membrane is extremely wrinkled, and to the point that, you know, in a vertical local curvature of this membrane, your proteins will be almost like parallel to your average surface plane, and you're losing that offset information there. So if in an extreme case the undulation is so high, then there will be a bias that will make me underestimate the measured protein height. But even in the GUV case that was not so taut, I didn't really observe the negative bias so far.
Thank you. Thanks a lot, Sungmin. So Christopher Kelly asked another question, partially answered. What was the area of the membrane that was averaged together for each height measurement? And does it actually matter that if the GUV, let's say, is 5 micrometer versus 20 micrometer, as long as you readily average?
Right. So I actually want to show another interesting aspect of this technique related to that question. I try to remain short. So the answer is no. So it doesn't matter how large or small this sphere is. I can measure the protein size the same way, with the caveat that the fluorophore you're tagging the membrane is not polarized. So this is actually the technical point that I spent so much time troubleshooting. Basically, this shows the opposite of my answer, where, if I change the size of the bead, my protein height measurement is actually changing. And later, I learned that this is not the actual fundamental aspect of this technique, but it's because of this artifact introduced by the fluorescence polarization. So if the dipole is systematically polarized in a spherical curvatures, it can introduce some offset because of this tilted 3D point spread function. And because of this, in some applications, polarization of dye is something we have to consider. And this polarization, it becomes more extreme as the curvature of your bead or cell becomes smaller and smaller. So we are seeing weird increasing height measurement here. But as long as you're choosing your fluorophore is not polarizing, this curvature shouldn't be an effect, and the height measurement should always be consistent regardless of that.
So in line with this, Sungmin, the localization of your reference probe is super important. And in one of your cartoons, you show that if the probe goes both leaflets, you are measuring the average height of the bilayer, right? So that's why you're picking probes that would go to one leaflet. Is that correct?
That's right. So for the measurement of the membrane surface, so usually protein tip is labeled, and I have to measure the surface of membrane. I usually use a very small peptide that is conjugated to fluorophore that directly sits on the surface of membrane. So in the real cells measurements, you can achieve this by using a toxin, for example. But something that is not polarizing should be the standard of the surface.
And also you mentioned the chromatic aberration, that you have the ways to correct it. What is the acceptable chromatic aberration range? Is there a limit that we can't recover anymore? It's too much.
That's a good question, because most of the experiment is done with the chromatically corrected lens with the Nikon 100X. I think one very important aspect is that aberration is consistent in the different locations of the field of view. So those transformation between two different colors, if you look at in the whole field of view, is pretty complex. But in the CSOP technique, because everything is axisymmetric, the only effect that this chromatic aberration is bringing in is different size of circles. And in the edges of your field of view, where chromatic aberration is shifting more, you still get get identical difference in the radius of those two circles. So that's because this aberration is linear, although the extent is different from the center to the side of the field. So as long as it's linear, I think the extent of the aberration itself is affecting less to the accuracy of the technique. And in most of cases, I would expect this aberration to be linear in the field of view.
Thanks, Sungmin. Next question is by Christina, and she's asking, could the height of the protein at the cell surface have an effect on the membrane fusion, similar to the effect that you see for phagocytosis?
So like I alluded in my main project in this lab, I've been actively investigating that possibility. So in the in vitro system where I use a synthetic membrane and decorating its surface with dense concentration proteins, the answer is yes. It seems like the energies required to fuse those two membranes together seems to increase significantly when there's a proteins in between compared to the bare surface. I've been testing this in vitro finding in the actual influenza fusion examples. And I still have to test this further, but preliminary data suggests that the natural protein, surface proteins is not bringing too significant of an energy barrier. But if you're decorating them with the antibodies, those such as neutralizing antibodies, those high concentration antibodies on the virus protein surface will bring in a significant energetic barrier diffusion, actually.
Thanks a lot. So the next question by the audience is by Eje. What role does the height of the glycan molecule play in the microenvironment that modulates the cancer cell? So you showed one single cancer cell, but would it have an effect on the tumor microenvironment of the tumor tissue? Can the height of the glycan be responsible, for instance, for the hardness of the tumor microenvironment?
For the hardness?
Of the tumor microenvironment.
Right. So I think the answer is yes. So in the tumor microenvironments, which has very different glycan profiles, such as charged sialic acids, there, on top of this crowding effect I'm seeing, there is this charge effect as well that will likely to make proteins repel to each other. And it also repels proteins from its underlying ECM, which is also decorated with other charges and small molecules. So I think it might be very interesting to apply this technique in the context of tumor microenvironment, although I can't think of exactly how you do it at the moment.
Thanks, Sungmin. By the way, we still have a few questions. Dan, do you have to go at 9 sharp?
Well, we do have a lab meeting at 9, so pretty close to that.
Okay. So maybe I'll ask one last question and then let you both go. And I will maybe type the questions later for you.
Yeah, we'd be happy to address them later time.
Yeah, so the last question, E-Catherine changes from floppy to rod-like conformation depending on calcium. Did you measure the height with or without calcium?
I did. I tried very hard to capture that difference. I do see a significant change with or without calcium because calcium is stiffening its interdomain linkers. But it wasn't as impressive than I would have anticipated from the other structural work that involved E-cadherin. And I think that's because there is other sources of flexibility to these proteins when it's in the dynamic states. And those stiffness effect to length might be dampened because I'm measuring the average height of E-cadherin while it's swinging back and forth. So I do see some change, not impressive enough to make it a headline of the paper.
Thanks so much. Thanks so much, both of you. I encourage other people whose questions that I didn't ask, they can email you. I hope you wouldn't mind. And
please do.
It was such a pleasure for me to have you both. And I hope to see you again soon.
Sounds great. Thank you again for the opportunity, and it's much appreciated.
True pleasure.
Thank you very much, Ardeen.
Have a good day.